We're at the 25th CROI 2018. We're here with Mary Kearney, who's from the National Cancer Institute, and I'll let you give more background to your affiliation as well as tell us, we just left the press conference, so tell us a little bit about yourself first.
Okay, so I'm in the HIV Dynamics and Replication Program in the National Cancer Institute in Frederick, Maryland. And the goals of my lab include understanding how HIV persists despite antiretroviral treatment, so despite ART. And so we've been studying for many years mechanisms that HIV is able to survive. And so there's sort of an ongoing controversy and two schools of thought of how HIV persists despite treatment. One of those schools of thought is that ART is not fully potent, and so you still have ongoing cycles of viral replication during treatment. The other school of thought is that ART is fully potent and there's no new infected cells during treatment, but those cells that were infected before treatment are able to persist despite ART, and they can persist for many years, most likely through clonal proliferation. So once a cell is infected before treatment, that cell can undergo cellular proliferation or clonal expansion, carrying the HIV provirus with it. So we did a study in 5 patients looking at these two possible mechanisms. And what we found is that whether you look in the blood of an individual or whether you look in the lymph nodes of these same individuals, it appears that there's no ongoing cycles of HIV replication, that the cells that persist on ART were already infected before starting treatment, and that they persist both in the lymph nodes and in the blood through cellular proliferation.
So how do you determine this? Can you give us the mechanism? How do you actually define this answer?
So what we did is we sequenced the proviruses, so that's the integrated HIV in cells, and we sequenced the proviruses in single cells in the blood, and we sequenced the proviruses in single cells in the lymph node, and then we compared those populations to each other, and we compared the population to the HIV that was there before starting treatment. And what we found is that the HIV population, the genetics of the population, didn't change at all. It didn't change from pre-therapy in the lymph node after many years on ART, and it didn't change in the blood. So it just suggests to us then that these cells were already infected before treatment and were able to persist. We also used another test called the integration sites assay, and we did this through our collaboration with Dr. Steve Hughes and Xiaolin Wu. And in that test, you can actually sequence the site of integration of the provirus in cells, and we found that cells in the lymph node and in the blood have statistically the same sites of integration, suggesting that these are actually the same cells in the lymph node and the blood. They just have, they're just siblings of each other. So they've just persisted through clonal or cellular proliferation, and these cells are being trafficked back and forth between the lymph node and the blood.
Right, and this is something now that as far as the next step, what would that be? Because you've kind of answered the question. I mean, you were put a few questions by some of the press about that you didn't have enough numbers or whatever, but you felt confident in your results as being able to would there be any reason to confirm any of this?
Well, I mean, certainly. So we've looked at only in the lymph now. So in the blood, comparing the cells, the infected cells in the blood from pre-therapy to on therapy, we've looked at a large number of patients, and we've published several studies, and we have not found that the population evolves in the blood. In the lymph node, we looked at only 5 patients, which seems small, but we found the consistent result in all 5 patients, that the population that's in the lymph node is exactly the same as the population in the blood. These cells are being well trafficked in all 5 of these patients, and so I wouldn't expect that you would find anything different in other individuals. Of course, it's always useful to do larger studies, and that should be done. But getting lymph node samples from patients on ART is difficult, and these, this is really a gift that they give because the patients are not benefiting from this in any way. They're doing it purely for the good of science, and so it's difficult to obtain those kinds of samples and to find patients who are willing to do that. And there are risks associated with donating lymph nodes, although now they have new ways of doing it that have really reduced those risks significantly.
It's like a little excision somehow.
So, but I think that because we found the same result across all 5 patients, that I think 5 is probably a sufficient number. But of course, it'd be great to look in more. Now, just because we didn't find any evidence for ongoing replication in the lymph nodes, and we have not in the blood, it doesn't mean that there aren't other sites in the body where ART may not be fully penetrating,
So
and
that
that
might
you
be the
could
next
have
study, yeah.
right. So you could have ongoing replication in these sites is possible, and so some further studies of this leads then to look in the gut. I mean, is this consistent in the gut? If you look at the T cells in the gut, is there any evidence for new infected cells there, or how about in the brain? And a lot of those samples, a lot of those studies can be done in animals as well, because it's difficult to obtain samples from patients from all these different anatomic compartments. But then it leads to assuming that it's consistent in the gut and in the brain and in other anatomic sites, that there's no evidence for ongoing replication, and then we conclude that ART is fully potent. Then it leads us to developing strategies towards a cure that target these cells that were already infected before starting treatment and are persisting through clonal proliferation. So we would have to target those with specific drugs or antibodies or something that can detect these cells and kill these infected cells to cure HIV. So that would be the next step. And there are many people doing these types of studies already, looking at with the latency reversing
anticipating
agents.
that this is happening. You've confirmed a piece that okay, now we really feel confident that we can move forward in this direction, and we'll get some answer that we can count on. So Dr. Miller's bringing out that subject, I think, about the brain, and it's I think we all worry about that, because seems to be a rather crossing the blood-brain barrier is something that's very challenging and concerning for having ART that really does the job there.
Right, and right. So I mean, I would imagine that if you have ongoing replication, probably anywhere where ART is not penetrating, that you would get drug resistance eventually, and that you would fail due to drug resistance, and we're not seeing that. I mean, typically where we see people failing with drug resistance is because they either had transmission of a drug-resistant variant, or they haven't been able to be sustained on treatment for one reason or another. So I imagine that maybe that's how we'll learn, if we start to see people failing with drug resistance very frequently, which I think we would have seen already, that maybe we would conclude that there would be ongoing replication, maybe someplace like in the brain. But my guess is that's probably not the case.
So this is a pretty important piece of knowledge that is in the building blocks that go towards a cure. And I think, and your work would your work expand on this knowledge, or would you, or what is your next?
Okay, so our next steps are to hopefully do some gut studies and see if we find in the gut the same that we found in the lymph nodes, and that is no evidence for ongoing HIV replication. I don't know what we'll find in the gut, but that'll be that's on our plans. We're also interested in looking at the efficacy of latency reversing agents. So we have an assay where we can look at HIV proviruses that are completely dormant or quiescent versus those that are actively transcribed and making HIV RNA and virus particles. So we want to look and see if you treat patients with a latency reversing agent, which should induce the transcription of these proviruses in order to then kill these cells. So we'll use this assay and we'll look at different latency reversing agents, and we can determine their efficacy and see if we can find, some latency reversing agents that are able to actually activate a broad spectrum of proviruses to kill these infected cells. So we hope to move we'll still continue our studies in characterizing the reservoir and understanding what the reservoir is, but we hope to move towards studies that are more interventional. So studies where we can actually try to target and disrupt or latency and maybe kill some of these infected cells and make progress towards a cure.
It seems like it's pretty important work that you've done, and you're one of 4 that were picked to do the press conference. I think not just because of your attractive personality, but you did good work.
Thank you. I don't think it's my personality, but
I
hope it's my work.
It's always important to have somebody.
Because you don't know me well enough. My personality didn't get me there.
It's always important to have somebody who can project.
Yeah.
And I think that's what I keep talking about the young investigators. They have to know how to project and present their case.
Right.
Because you're really arguing your case, which you did very well.
Yeah, thanks.
Well, thank you so much for being here.
Thanks for having me.